All right, so thanks to all of you for joining us at the first ever virtual Beckman Institute Directors Seminar. I'm Patty Jones. I am the Associate Director for Research and your MC for today. As you may know, the Director Seminar features either a faculty member or a combination of postdocs and students to give up-to-the-minute talks about all the great research that's happening at Beckman. And we're very pleased today for our first virtual seminar ever to also have two speakers who are going to talk about COVID-19 research. Each speaker will talk for about 15 or 20 minutes, and we'll have some time for questions. All of you in the audience do not have the capability of unmuting yourselves, so you must use the text chat to ask questions. So please put the questions in the text chat, and I will read them out at the end of the talk. So I'll be refereeing those questions for the speakers. So while we are still, wow, there's all these people coming in right now. So while we are still having people filter into the virtual room, here we go. I will turn the floor over to our first speaker, who is Nick Angelo, as you should all be able to see on his first slide. Nick is a graduate student in chemistry in Marty Burke's lab, and he is one of the main people building our new future core facility of the Molecule Maker Lab. And he will therefore be talking about a specific project related to that, as you can see from the slide, on synthesis and testing of COVID-19 binding peptides at the Molecule Maker Lab. Take it away, Nick.
Thank you, Patty. Let me just make sure this is all okay. Thank you, everyone, for coming. I'll start by stating the obvious. We're in the midst of a historic pandemic that has disrupted all aspects of our society and our university. As a community, I'm very proud to see that everyone has been coming together to tackle this, from scientists to public health officials to students. 6 months ago, I couldn't have imagined that we would be doing this kind of testing that we are now, with the tests made at Illinois and deployed at scale. So if any of you aren't familiar with this, this is the SHIELD testing data. This is like publicly accessible and shows all the tests we've been doing on campus, the total number, and then the rolling rate of recent positive cases. And so as a graduate student working on campus, this wasn't available a while ago, and so it makes me feel a lot safer about actually conducting work. Looking into the future, we need to make sure we're at an even better spot 6 months from now. So going back to the basic science of how COVID-19 affects human cells, it takes place through human angiotensin converting enzyme 2. So human ACE2, it's just a cell receptor. It's related to regulation of the blood. And what the coronavirus does is it has these spike proteins on its surface, and those hijack ACE2 to bind to it and allow it to infiltrate the cell. And so the right structure here is a crystal structure of the receptor binding domain of the coronavirus with human ACE2. And highlighted in blue are two alpha helices, which we hypothesize we could make minimized peptides from, corresponding to those alpha helices shown in blue, and then that could serve as a binding motif to the virus for a next generation COVID-19 test. So there's a lot of reasons that we think this may be the case. There's a recent study by the Procco lab at UIUC, which used deep mutagenesis, a kind of high throughput biological technique to mutate all the residues of ACE2 and see which expressed under the selective pressure of binding to RBD. All the blue squares on this checkerboard in the bottom left are hotspots for mutations that increase binding, and they're almost all in those first two alpha helices. They also went ahead and isolated one of these mutants from a biological mixture and showed that it had better binding than the wild type, so stronger binding, and also could outcompete it. And they're trying to develop soluble versions of this for therapeutics in the future. There's also recently emergency approved testing card costing $5 and taking about 15 minutes from Abbott Labs. This is great progress, but we still need a test that's as inexpensive as this test while being sensitive enough for early detection and being able to be taken at home before someone has to enter their community. And we hope that in 6 months we're somewhere near there. So this is the layout of the talk. There's 3 little sections, and it's all very collaborative. So the first is the computational design of ACE2 peptides. So that's trying to understand and design computationally better binding peptides is then synthesized in the lab, and that will be done through automated synthesis at the Molecule Maker Lab. And then finally, there's some very exciting work by Jing Wang and his colleagues about making designer DNA nanostructures that can bind to the surface of the coronavirus and kind of push the limit of detection. And we hope that by designing peptides that can bind the coronavirus very, very well, and then combining it with this DNA templating strategy, that we could potentially design an at-home test that's also very sensitive and also very inexpensive. So at the Molecule Maker Lab, we're really we're a maker lab. We're trying to create this facility where people can think about small molecules or biological molecules like peptides or oligosaccharides, just as they would at any other type of maker lab. So we want people to be able to learn about molecules, design them just like they would build stuff with Legos, build them on an automated synthesizer, test them in their desired application, and then share their experience with the world. And so practically, we've been building this lab for about a year now, and practically it's a room with a lot of automated machines. But there's a lot of power in that, because these machines, they put together these different types of building blocks like peptides and oligonucleotides and oligosaccharides, and even some small molecules, and they put them together in the same way. And so they could be very, very specialized to putting them together in a way that works and is very efficient. So today's story is just going to center around polypeptides, but there's a lot of other little stories like this that are going on. So to start with the computation. So Ahmad Tazkhurshid and colleagues are working to make a computational workflow to target these ACE2 spike protein surface and interface. And so shown in red is the receptor binding domain for the coronavirus, and then shown in blue is the ACE2 peptide, with the gray underneath it being the rest of the ACE2. And I'll just click to start the little movie, just to show these are molecular dynamics simulations. So we're looking at how these molecules bump into each other effectively and see how they bind. And so the idea here is that we design the derivatives computationally and check the stability by doing these molecular dynamics simulations. And if they are stable, then we go on to complex them with the S protein of the receptor binding domain and see if that's stable and measure the binding affinity, and then we can rank them. So to give a couple more nice pictures, there's a zoom in of these two alpha helices of the ACE2 peptide, and some of the specific interactions which we can observe between the receptor binding domain. And this is specifically the residues 18 to 84, so it's 66 amino acids long. First key finding is that in explicit water, in molecular dynamics simulations on the 100 nanosecond scale, it appears to both fold into the desired conformation and also be stable. And then going a little further, complexing that structure, just the native wild type to receptor binding domain, appears to also be stable in these types of simulations. So that means that using this workflow I described, our colleagues could mutate specific residues and see how it relates to the binding. And so that gave us a hit list of 16 mutant peptides which show higher binding, and gratifyingly, all these complexes were found to be stable when subjected to these simulations. So to show that another way, there's different amounts of hydrogen bonds between the wild type shown in red and these mutants. Some have more, some have the same. And some of these mutants, which are lower in energy, so more stable binding, have either more or less contacts than the wild type as well. And so what does that really mean? I like these sets of pictures a lot. So this shows that as a network analysis, if you mutate the ACE2 peptide, then there could be a lot more interactions with the receptor binding domain, which could be the cause of its stability. And so all these kind of lines in between are showing correlated motion. So during those dynamic simulations where the receptor binding domain is wiggling and the peptide are wiggling, these areas correlate. And so couple dynamics that shows some stability. So on to actually synthesizing the peptide. So we use iterative peptide coupling on an automated machine. So the way that works is pretty simple. you have an unprotected carboxylic acid terminus on a bifunctionally protected block. So the amino acid, like the amine, is protected with an FMOC group. You then react it with amine on the left side to make a block that's grown by one, and then you selectively deprotect and reform the amine to then repeat this process. And so we use a Liberty Blue microwave peptide synthesizer, and this instrument uses microwaves to accelerate the coupling. And so it gets to about 90 degrees within 30 seconds, and then it takes about 2 minutes to form an amide bond. And so there's a lot of work we did to make this work as well as possible, but I'll just tell you about a couple different aspects. So first thing to know about peptide synthesis is that it's done backwards. So this is the sequence at the top, where it starts with S and ends with P. And so when you're doing synthesis, it's done backwards. So starting with proline, it has this side reaction which can happen with any amino acid, which is basically the second amino acid getting on this growing chain can kick out the first one and make this cyclized structure, this dichylpiperazine. So that's not usually a problem because it's kind of slow. But once you have proline, which is the first amino acid we have to start with, that 5-membered ring gives it some tight bond angles, and that allows it to more easily perform this reaction. And so when we're making a peptide this long, we would have preferred to have a relatively unhindered and low loading resin such as this MPA resin in the bottom right. But what we ended up having to use is this very hindered and higher loading resin, TCP chloride resin in the middle to prevent this reaction. And the second aspect is how we actually perform the synthesis. So I'm showing you 2 snippets of the actual code. So it's as simple as typing in the amino acid sequence and then picking which types of couplings are being done and then running it. And so I'm showing 2 sets of conditions we tried. And usually when peptides get this long, about over 30 amino acids, they might take some amount of optimization. But we found that by just doing a single coupling, which is just a normal amount of reagents and not an extended amount of time or not an excess amount of reagents, we got equal results to having doubled the amount of reagents and cycles for the last half. And so we have pretty high confidence that this is actually relatively optimized once we got all the right reagents and components. As Linda described a little bit of what we actually do with it after we plug it in on the machine. We take it off and we submit it to a cocktail of acid and a couple other groups which kind of pull off all the protecting groups. And we precipitate it in ether and then rinse it for a few times and then dry it. And after that, you can tell that in the crude mixture, there's a lot of these low molecular weight organics in the mass spectrum. But after the purification, it's pretty much entirely these multiply charged peptides fragments showing up in the ESI mass spectrum. And so just a word on the purification. We do preparative HPLC and it ends up being a fluffy white solid at the end. And this was a little bit out of our wheelhouse because I personally hadn't made peptides like this before. And in a lot of our initial trials, it came out as about a 10-minute band, which is not ideal and was kind of difficult to understand. But after some optimization, we got it to come out as a tight peak that looks very, very pure by every method that we have so far. Okay, so what are we about to do with it? So we're going to use surface plasmon resonance. It's a technique to study binding. So to kind of paint a picture here, we have a gold surface and we have these blue Ys, which are the receptor binding domain of the virus adhered to the surface. And what happens in SPR is a light shines at the gold surface and there's a specific angle that reflects back called the resonance angle. And that is the angle at which the gold electrons are resonating. And so there's less signal there. It's a dark band. And so once you add something, in our case, the peptide we're making, a binding event is seen, and then that shifts the angle, and then that's how you measure it. And so as far as I'm aware, this instrument was improved and is now ready to use and we have some experiments planned early next week to try to validate the peptides that we're making and see how they compare to, for example, the full ACE2 peptide. And SPR can vary a little based on how you have it set up, but that should give us a good context. The final story comes out of Xing Wang's lab, one of our collaborators, and this, I'll start out with a little recent success published in Nature Chemistry in 2020, where the dengue virus was selectively bound to with this DNA nanostructure star aptamer. And so the surface of the dengue virus is very complex, and there's a lot of aspects to it. But thankfully, it is a little rigid. And so by doing DNA origami in a very specific way, there can be multiple sites which the DNA can bind to on the surface, and that linked up with a fluorescence signal can make and did make a dengue test, which is higher limit detection and cheaper than anything else on the market. And so, so this is just showing the data of it being better limit detection, especially can detect even before the onset of symptoms. And so compared to PCR, just like we do with the coronavirus, it's actually better in this case, and also the same with ELISA, which are the gold standards of what we do nowadays. More interestingly, just to kind of show the structural importance here, so you can make a whole variety of shapes with DNA origami, but in this case, like matching the surface of the virus is really important. And so the star construct is about 7,500 times better at binding to the surface of the dengue virus than this aptamer. And this type of binding is very important because there's a translation problem in science when these types of small molecules or biomolecules are found that can bind to specific proteins in a virus or in a cell, where the fact that there's all those other proteins surrounding it in the cell or in the virus, that actually like drives the binding significantly down. So having a kind of putting this problem out front and designing a DNA structure that could bind to the coronavirus's like viral outer shell is kind of a key strategy we're trying. And so the state of the art of that right now is under development, but I think actually moving pretty well. And so the coronavirus has a surface speckled with all these spike proteins, and they're trimeric typically, and those are what binds to ACE2, and then what allows it to proliferate in the cells. So the coronavirus is a little more functional than dengue, and so it's not 100% known whether the strategy will work, but it seems very promising that it is, because even though it's somewhat functional, you can still derive a basic pattern that some DNA origami can be done. And so I think that data and that strategy is close to being, out there in the public. And so with that, I'd like to thank you all for listening to my brief talk, and I'd be happy to take any questions. I'd like to also thank the people on the left here. This is the COVID-19 peptide team. So it's Marty, Jeff, Ahmad, Xing, and also Beth, who's helping us with the SPR. And then my colleagues, Arun and Shashank, who helped with the synthesis and the modeling. And then on the right here, I'll just briefly mention the Molecule Maker Lab. Like, we've been building this for about a year, but happy to say it's now been funded as a AI institute, or as part of an AI institute across multiple campuses. And so I think just keep an eye out for that. Hopefully we'll be doing a lot of good stuff there. So yeah, happy to take any questions.
All right, great. Thank you, Nick. I do have some questions here in the chat. First question relates to slide number 12. How did you choose the type of mutagenesis to perform? Was that a single mutation or multiple mutations at a time?
Okay, so I believe I'm sure they started with one at a time. And I'm sure it was, as far as I know, it was guided by the work that Eric Proko had done before. And so it's no problem for us as synthetic chemists and using this machine to change multiple at a time. But I think from a combinatorial standpoint, it'd be like many, many more combinations to do that. So from my understanding, it's one at a time. But there are some that have two.
Okay, next question. In considering therapeutic applications of these peptides, how many of these ACE2 peptides must bind to a different receptor binding domains of the COVID virus to prevent cellular uptake?
So that was an excellent question. But I would say that generally at this point, I don't think it's very well known exactly what would be required. So I would say this is definitely to be discovered.
Another question is it practical to use a synthetic peptide this long in a diagnostic test?
I think so. I mean, the yield's actually, so it's surprising, right? So when you're doing it, at least, okay, so to scale up, potentially like there's a lot of very creative, easy ways to solve this problem. If this was like a real problem. And so theoretically you could make half, then put half together, something like that, and do those on larger scale. But it's surprising that it even works really well at all, at least to me as just a traditional synthetic chemist, because we're doing 66 steps here and we're getting like a relatively decent yield. So that's, yield is multiplicative. So it has to be basically, it's pushing the limits of being nearly 99, 100% each type of reaction. So it seems practical, but it really will depend on how miniaturized we can make the sample chips. And I think we're pretty far away from seeing that, but also, I think it's a pretty exciting space.
Another question is, what steps are needed to build a preliminary Wang-type DNA device as soon as possible and check for sensitivity and specificity for binding to COVID-19? And how will the device read out the result?
Yeah, that's another good question. I think that is also still relatively up in the air. So I think for fluorescence is actually less like, there's a lot of hard aspects of designing an at-home test, I would say. And you can't just have it be a fluorescence readout because people don't have a fluorimeter. So it have to be something visible or something just like that test I showed earlier, where it's kind of like a pregnancy test, where basically you have an obvious visual indicator. Yeah, the steps. What are the steps to get there? We need to get peptides, get SPR, then know which ones bind, and basically how much peptide we would need. So a practical consideration like the other person brought up earlier. And then the DNA nanostructure, we need to attach it to the peptide. There's a lot of really good chemistry for doing that, but we haven't necessarily done that yet. And then I would have to, as far as the state of development of Jing's nanostructure right now, I would say you'd have to ask him, because I think they're working in the industry to kind of try to make it a reality faster than I can disclose, I guess.
Another question is, are there plans to show stability of the two helices, such as?
Yes, that's also a great question. So, and I didn't bring this up either, but yes, definitely. So everything we do to the peptide, it's going through an entire hot synthesis. So at the end of it, like, is it even the conformation we want, and so on. And we think it should fold, and everything is pointing towards it will fold, but it's definitely worth exploring through, again, pretty simple means like circular dichromism. So we will explore that, and if it isn't doing what we want it to do, there's pretty classic refolding techniques which can be done. And once we have everything is a lot easier once you have a really pure peptide. And so now that we're there, I think this is not a huge barrier.
Another question, how large of a peptide library can you practically synthesize or handle for the experiments relative to the computational models?
So I really should have brought this up too. So with our optimized procedure, we are doing 66 reactions in a row. However, they're so fast that it's about 6 hours to make one. And so we have 16, our hit list is 16 right now. And so I'm not going to say we're keeping up because we don't have all 16 and we haven't tested all 16. But it's not any difficulty at all, really, to set up the synthesizer to run for a few days and make, let's say, 12 or more. So we could do batches like these. So I think it's actually pretty practical, and for at least learning the rules, because if you look at this this data, which is still up here, some of them seem to be key winners and some of them seem to be key losers. And the faster we find that out, the better, I think.
All right, and one more question is, do we need a higher affinity peptide, or could the natural ACE2 peptide be enough given the polyvalency effect of the Wang-type DNA nanostructure?
So the answer is maybe, but I think the problem with the full ACE2 peptide is okay, so this is for in terms of testing. In terms of testing, it's possible. It would work fine with the ACE2 peptide, unless there's some size effect preventing the polyvalency from working, which I know is the case, but depending on the tether we put on it, it shouldn't really be a big problem. But as a practical consideration, we can get, tens, hundreds of milligram quantities of this peptide, whereas as far as I'm aware, purifying the full ACE2 peptide has to be expressed in a cell. Then it's kind of a much harder industrial type process, I would say.
All right, I think I'm going to cut off the questions there. So thank you so much. Let's give Nick a virtual round of applause by your little reactions or
Thanks, Patty.
actually clapping on your video feed. Thanks very much. All right, so thank you, Nick. Let's switch over to our next speaker, who is postdoc Mayank Garg. He's a postdoc in the Autonomous Materials Systems group at Beckman. He's working on building self-healing and self-cooling structures for aviation, automotive, and space applications using FROMP, that is frontal polymerization. He earned his PhD here at Illinois under the guidance of Professor Nancy Sotos in the Department of Materials Science and Engineering, where he developed various sacrificial polymers for enabling rapid energy efficient manufacturing of microvascular polymers and composites in multifunctional applications. And as you can see, the title of his talk today is Adapting Design Thinking to Existing Supply Chains, Rapid COVID Testing. So take it away, Mayank, and again, audience, please use the text chat to send me questions, which I will ask Mayank at the end of his talk. Take it away, Mayank.
All right, thanks for the introduction, Patty. Good afternoon, everyone. Today, compared to Nick's talk, which was highly technical, I'll be talking about slightly different aspects of two different projects that we came across during the lockdown, where we started using existing supply chains and materials to ramp up testing in the local community. And before I go into the actual projects, I'll start off with some brief motivation, and I'm not sure why my slideshow is not working. I don't know if you guys can see my screen. Okay.
Yeah, I can see it. Yep.
Yeah,
it's fine. Right.
So before we started working on these projects, my roommate and I, as a part of his celebration after his prelim, were watching this movie called Padman, where the guy in the picture was able to make sanitary pads accessible to millions of women in a cost-effective way by just simply miniaturizing the existing methods and the raw materials and the machines and deploying them into local villages to keep the economy of the profits of the product in the community, and that thereby reach millions of people. And it's crises like these which have existed before COVID, which still motivate how we can tackle emergencies like COVID. So this movie got us thinking into this an interesting discussion of how sometimes fragmentation of the manufacturing is actually better to reach out to local communities rather than looking at a one-standard solution from the top. And that became evident when this request from the governor's office came on April 5th, where there was a shortage of COVID-19 testing swabs, which are these highly engineered swabs which are disposable for single-use COVID testing, and they are put into the nasal cavity. And these swabs are supplied by very few suppliers, and they consist of two main components. One is the rigid handle with the flexible stalk, so that you can actually reach deep into the nasal cavity and bend the swab easily without discomforting the user too much. And the second is this flock region, which can collect the mucus effectively with this soft brush fabric. And there's very few suppliers available, and these were in short supply, and the cost of these materials were driving up. So the governor's office has a target for the state of Illinois to reach 10,000 tests per day, and they were at 5,000 at that time. And they needed to find ways to use completely synthetic materials which can be sterilized and manufactured in a cost-effective way in millions of units. And the way industry answered that call was by designing using 3D printing techniques. These highly sophisticated 3D printers were used to create swabs. And contrary to the traditional Copan swab, which was two components, this is a single material construction, but the head of these swabs is made in such a way that it can bend around easily, even though the handle remains rigid. And there were some recent studies published in a journal where they discussed the qualitative as well as the quantitative comparisons between different swabs and what kind of design parameters are essential related to sample collection as well as the bending and the rigidity of these structures. But these swabs at that time in April still remained inaccessible pending FDA approval and very high cost. So the request came to IUC saying, can we 3D print them in our lab, in our university? And most academic universities now have a lot of 3D printers. So the natural question was, oh, can we simply just use our existing printers to do this process? But then there's questions like, what are the kind of raw materials we need to use? What kind of technology do we have it? Do we have all the equipment? And can we even scale it up even if we had one specific equipment for this purpose? And it became clear that this would be a tricky question in terms of using a 3D printing resource from a university to scale up manufacturing. And the mantle was passed on to the Autonomous Materials Systems group at Beckman, because we are experts in actually manufacturing different types of plastics very rapidly using the concept of frontal polymerization. So the director, Jeff Moore, reached out to, contrary to reaching out to chemists, he believed that this is a problem for engineering people. So he reached out to the engineers in our group to figure out a way to reverse engineer these COVID testing swabs. And through their discussion over email, it became clear to them that 3D printing was not the way to go based on the equipment we had. And maybe we should try casting molds where we can make a soft mold and we can use frontal polymerization to create these thermoset swabs rapidly using that idea. Until that time, I was not part of the equation because I was not part of the email. So I had no idea that this was happening. But just sheer luck or serendipity, Jeff, just the day when they wanted to go into the lab, reached out to me on Twitter saying, hey, we're going to make swabs. If you are available and comfortable, please come. And I really wanted to help. So I joined the team. And we went in with our initial idea of trying to make a mold for these swabs. And it quickly proved to be not a feasible option because of several reasons, including the manufacturing itself, where we were not able to easily replicate that flock side. And there were some challenges along with how to prepare that many molds and how to train people and get the FDA approval for such raw materials that we would be using. And then this subconscious inspiration from the movie kicked in my mind, where I started thinking, why do we need to start from bottom up? And the bottom up approach was shown in the first presentation, where it's really important to do the fundamental side. But in this particular example, I felt that we need to start with a better material to put us into a position where we can create a minimum viable prototype. And that's where I realized that I had these microelectronic swabs sitting in my drawers for a few years because I was using them in a particular project. And these are highly precise, clean room grade microelectronics fabrication swabs. They're extremely cheap compared to anything medical grade available. And they are part of a non-competing supply chain because nobody's interested in them right now as far as COVID testing supplies go. And they are made from polypropylene as well as polyester materials. And the head is a knitted polyester, which made me think that this should absorb the mucus of the sample really well. And if we compare that to the Copan swab, there's one main challenge though, that the length of the swabs that I had in the lab was almost half of what we need. And we need that full length because the practitioner needs to be at a safe distance and reach deep into the cavity of the nasal cavity of the person to collect the sample. So these electronic swabs were too short and stiff. First of all, they were not as flexible in their current form. And there was no clinical validation for these materials, even though we thought it should be easier to do that because of the high quality control on producing these kind of swabs. And any polymer engineer in the audience will tell you, well, if the shape and the length are not right, why don't you just simply, it's a polypropylene material, it's a thermoplastic, why don't you heat it up and stretch it into the desired form? And that's usually, it's easy to do that for fibers in industry because they have sophisticated equipment to do this process very easily in a continuous fashion. But for a lab like ours, which does not have a drawing machine, we came up with our own ingenious idea of using binder clips, weights, tapes, and ovens. So we had these swabs sitting in, and we figured that whatever we have available, we need to use that to our advantage and draw these swabs individually. So that's where the concept came, where we could simply clamp the swab between two binder clips and put the weights on the bottom of the clip and then heat it into the oven for a few minutes so that the polypropylene can soften. And then just like yoga, let gravity do its own thing and then take care of the rest of the stretching part there itself. And within a few hours of iteration and testing different conditions and different sample types, we got to a working prototype where we could actually, sorry, I think it's loading. Can everybody see my presentation?
We're back. Oh, here we go. Back to 0 to 1 slide.
Okay, yeah. So
Okay.
within a couple of hours, we were able to get our first prototype of the desired length and the flexibility that we wanted. And I really have to thank Dr. Doug Mitchell from chemistry, who had the courage to test this first swab on himself using with the help of a trained practitioner. And the feedback we got from this first day was that the flexibility and the length look decent, and the collection efficiency is working well in terms of the amount of sample that they need. And that's important because compared to usual training where we try to get every step perfect from one to the other before we get original feedback in the field, in this case we had to go from that 0 to 1 state in the first day to make sure we can get people excited and we can get the real feedback from the field, and then fine tune each of our individual steps later on. And that's what we got working on. We started, and this was undertaken by, especially by Doug and Gia, who came up with the ingenious ideas of using different multiple binder clips, hex nuts, whatever was available in the labs or in the Home Depot. They gathered that together and figured out a way to ramp up the production of these swabs to 4 to 5 each cycle. And then from there on, we started reproducing these swabs in a much better way. And so we were able to get the reproducibility aspect figured out, and then we were also able to start having a training procedure set up, so that with our original plan of using multiple volunteers, where we can actually have different stations across the university with ovens and binder clips and all those things, and we can start hitting our target of getting these swabs mass produced in a nice fashion. So with the help of Steve Drake, we were able to create a video for this process, and we were hitting about 10,000 swabs per station per week, which is still not enough, but it was much better position than what we had within a few days. And from there, we moved on, and this project started taking some attention from the authorities in the university as well as beyond. So IDPH, in one of the meetings, the Illinois Department of Public Health reached out to us saying, hey, we will be willing to do a clinical validation if you guys can send us 500 swabs for testing. And we had the existing supply chain, but we didn't have all these swabs on hand. So we reached out to multiple groups and clean room representatives from the university. Unfortunately, nobody had the right swab. So we had to get them ordered quickly. And within a day of staggered shifts and a lot of manual labor, we were able to ship them out to IDPH within a couple of days. And while we waited for the IDPH procedure for the clinical validation, we also started thinking of how we're going to sterilize and individually pack these swabs. With the help of Dan from Systems Purchasing, we came into contact with Stratus and other packaging suppliers, where they were able to show that they can use existing sterilizing protocols to individually pack these swabs into single units. And by this time, we were in a good position where we had enough materials to do the production, and our design criteria for the swabs were also in a good shape. But during all this time, we also realized after producing multiple samples that the procedure was quite labor intensive and involved, and each cycle would produce only 4 to 5 swabs every few minutes, and that might not be enough in the long term. And that's when, because we reached out to the university, we were contacted by Professor Placid and his grad student Nick Toombs, who work in automation, and they started looking at our training videos, and they said that, hey, we can identify some of the slow steps, we can try to figure out a way to navigate that and remove them. And Faraz Arastu, who's my roommate and who so far only in the first 10 days was mostly driving me around in all these locations to get supplies and to ship things out, said, hey, I'd like to be involved in this project as well. And he's one of those people who loves to automate things at home and at lab, so I thought he would be the perfect fit. And together they came up with this design of a trapezoid-like pizza oven, where you could feed swabs from one end, and then the heat would be applied and they would be stretched on the other end continuously. And they discussed this idea, and they realized that building it in a reasonable amount of time, in terms of a week's lead time, would be challenging and we need fine tuning. So let's agree on a realistic design. And I really have to thank Faraz, Nick, and Gia for this, where they had never worked together, but they came together on the first day and came up with all the constraints and design criteria for what's realistic, and came up with a very simple final design, where they decided that you can just stack all these swabs in this metallic clamp, and instead of using a binder clip, you can just clamp them together, put them into the oven, and do this process much more faster, because that was the slow step of loading and unloading. And within the third week of starting this project, we were able to have that final clamp with minimum cost, minimum spending in the machine shop, and we were able to get much more throughput and consistency. We improved our manufacturing speed at every location by almost 10 times, hitting the target of 100K swabs per week, which put us in a good position to now mass manufacture them if need arises. And by that time, we unfortunately never heard back from IDPH, but Carl had been participating in some of this discussion. And during this time, Ali from the Foundational Hospital reached out and said, hey, we tried your swabs on 10 patients, and the feedback we have is we're getting enough sample and we don't require any specific training because the materials are very similar. The question is, they're still a little too bendy. Can you fix that bending issue and make them more rigid so that the practitioner feels more comfortable when they're trying to collect the sample inside the nose of the person? And that's where this closed loop feedback was very helpful, because now we could go back to our initial design choice and manipulate one factor at a time and go and get this problem fixed. And since we had ordered these swabs from a catalog, they had a whole range of choices compared to the old swab, which is the thin stock. We also had the company had these thicker handle available with the same length and similar features, which by simply then choosing a thicker diameter and drawing that, we decided that we can actually have much more stiffness in the swab. And that was indeed true. Initially, we had to move through different again iteration cycles in the binder clip system for me to initially get the conditions down for getting these swabs to the right length and the right form factor. And eventually we moved that down into our giant clamp, and we figured out that now the collection, the sterilization, and the stiffness was all good as far as feedback from Carl is concerned. And they were looking forward to starting a process for clinical approval. And while we waited for a couple of weeks for Carl to respond, Nick and Faraz really took this whole thing one step further. Day and night, they built this horizontal drawing system. Instead of using ovens and a non-linear process, they figured out that we can just build our own linear actuation system where you can just load the swabs onto a clamp and move these rails, and they get heated only selectively in one location, and you can draw them much more uniformly. And they had very promising results to replace the oven system. And we were waiting on some communication from Carl, but by that time the infection rates were, I think, a little bit more under control, and some of the 3D printed swabs had gotten FDA approval. So we came on an official halt by the end of May, where we had a solution that existed in our lab, and that was shelved in case the need arises. We can deliver that. And that's one of the wonderful things about this project, that going in, we knew that we would really love to succeed, but we would hope that we would never need that solution if we can control the problem on the root end itself. But the lessons learned here definitely did not go to a waste, because once this saliva test was developed, which is a wonderful achievement, we were giving the saliva test early in the summer. And I think most of you would recall when initially you guys started, you would put your saliva in a tube, and then that tube, another person would put that into a separate Ziploc bag that would enter a 24-well rack. And then this would be taken to a processing facility where someone would unbag these, remove the sample, load them onto a stage, which can be then handled with a robot. And I remember joking to my roommate in the first week that UIUC must have some really efficient bag openers to make sure you can do this process really fast. And at that time, the testing speed was around 5K tests per day or so. But that joke became a reality in terms of the hardships that they had in this process, because I think on August 22nd, there was an email sent to Jeff saying, hey, we need some help trying to design a new rack system to overcome this problem and eliminate these bagging steps, so that we can direct the user or the sample provider can directly put these funnels onto a rack, which can go into a robotic system directly. And the main concept that was described to me on a phone call on Saturday by Jeff, quickly within a minute, was we have a problem where we want to connect these 24-well racks into a 96-well system. So we need to combine 4 racks on their long edge. And the question was again, can we 3D print these certain hinges or certain supports which can be put onto these racks individually, and then you can do that process. And my roommate was not available during the time, so I called Nil Parikh, who's a 3D printing expert and a composite expert in our group. I said, hey, Nil, I have a problem, and it will probably not help in your research, but it would be interesting to solve if you have some time. And he said, I'm in Beckman switching out samples, so one of the most boring stuff that I'm doing. Why don't you come here and we can try to figure it out? So the Mighty Clip won again, because we discussed that 3D printing was not a way to go in terms of thinking of how we have to design, iterate, and refine our structures to get them to integrate. And the binder clip solution just proved out to be more effective. We could simply take 12 of these mini binder clips, put them onto these 4 racks in different locations, and they could allow you to hold 10 times the weight of the rack itself, which is more than enough for this process. And it comes back to, again, the choice of design thinking again, that usually you would want to 3D print things because it's more cool. But at the same time, you have to start thinking that there's a scalable, cheap solution which is made out of metal, which means you can put this in boiling water for 95 C 30 minutes, which will be done every time you test these, you use these racks. that can survive and do the job for you. And that's when the biggest scavenger hunt in the campus or in the town began, where my roommate diligently drove me around in every single store in this town to get 6,000 clips, to procure them from every single possible location in every single color, with this, but keeping the size same, because that was the size that worked for our application. And it was challenging but interesting to get 5 or 6 volunteers from the AMS group on a Saturday afternoon to say, hey, we'd like to do a very mechanical, monotonous job of just assembling small racks into a bigger rack. But the kind of difference this made is you can actually increase I was told by Tim from the testing facility that they were able to increase the testing speed by 3 times using this solution for a temporary in a temporary form. And these clips are still performing if you see them when you give your tests. And they were simply there to buy enough time for a more permanent solution. And that's where I want to share my outlook about what I learned from these 2 projects, is that sometimes as grad students, we get stuck into this loop of we have to get everything perfect in the first go, step by step. That is not the way to think sometimes in an emergency, because you need something that we can put out in the field, get some real-time feedback, and then decide which parameters you need to change. And some solutions are completely sacrificial because you want them to exist only to buy time to get a more permanent one. So you have to understand that constraint. Instead of looking like in terms of forest and the trees, you have to think like, how far do I want to go with this first solution versus the eventual solution? And there's a lot of intrinsic motivation that drove this team together. It included a lot of friends. That was part of the success that we were able to achieve here. And it also involves things like getting support from the team members to make sure we can deliver on time. Sometimes we would have to stick around at 12 in the midnight or 1 in the night to make sure we can get a prototype delivered to Carl at 8 a.m. and get the real-time feedback, because that was way more useful than spending too much time perfecting every single step. And with that, I want to thank the SWABS team, the Autonomous Material Systems group, and all of my collaborators and advisors, because only with their support and their diligence, I was able to participate in this project. I was in the final year of my PhD. I was beginning to write my thesis, but the only reason I was able to tackle and spend time on this project is because my advisors were very supportive and my collaborators took some of the work that I was doing to make sure I can spend time and devote time to this wonderful, wonderful project. And I want to leave you guys at the last thought that resonated with me throughout this project, that you go to a war with what you have, not with what you need. Thank you. I'll be happy to take any questions.
All right, thank you, and thanks again for virtual claps or video claps to Mayank. And again, a bunch of woohoos and prompts and that kind of stuff in the chat to say that this was like a great partnership and so impactful and really helping the campus rise to the occasion of the testing it needed. A couple of questions along the way here were, one was, I would have thought that you couldn't even start on this until you knew you had like a clinically appropriate material to use, but you had started with those micro swabs. And so that turned out to be actually not a real big constraint.
Yes. So I believe at some point the FDA had allowed some sort of emergency use authorization. So they had put together that, like, if you can prove that this kind of any of those diagnostic tools are effective, you could bypass some of the stringent and time-consuming steps, and that was part of the motivation in the starting to make sure we can still push through some of these swabs. But we still needed a proper clinical validation to confirm like a proper comparison with the gold standard, which is the Copan swab during the time, to make sure we can deploy that to the mass scale.
Another question is, in terms of the human testing of your swabs, were you worried about that flexible end, like coming off and getting stuck in somebody's nose?
Right. So before we gave these swabs, we did a bunch of qualitative tests. We were trying to bend around the swabs, around different radii, trying to press on them, trying to figure out which swabs look good enough that they won't snap off easily. And this material was pretty durable, so we did not have that issue reported in any of our uses, which was great. But there was obviously some bad swabs which we were able to draw and remove them from the chain when we were trying to get the prototype running. And that was the whole motivation for coming up with that bigger clamp system, so that you can actually reproduce that quality control much better compared to the binder clip starting point. And I do want to point out that like one thing that I forgot to mention is usually we say in a seminar that, hey, please don't take pictures or this work is not published. I would love to see these ideas go through and be refined by other people. And maybe we can get these testing delivered to multiple locations in which testing is not really readily available so easily. So we need to come up with other solutions to reach out to such locations across the globe.
I think I saw on the state of Illinois' COVID page that there's like, I don't know, 8 testing sites in the whole state, and then there's like 17 on our campus,
Right.
So I think that, yeah, as we want to scale up testing, that's definitely a need. And I think, yeah, all of what you just said would be a fantastic kind of article in a technical magazine or, ASME or I'm not sure what the equivalent of Popular Mechanics is, but places like that where it's going to reach a lot of people. I mean, this is a wonderful story. So again, we are almost out of time, so I'll pause there. Thank you again to both Nick and Mayank for giving us wonderful talks about the inspirational and really cool things that that Beckman has done related to COVID. Again, you feel free to email our speakers personally if you'd like to follow up with them. We will have an edited version of this video and audio recording done at some point, and we can talk more with our communications folks about distribution of this. I'm not sure if it's going to be edited to be on our YouTube channel or how we're going to do that. And then our next director seminar will be October 8th, featuring Professor Chen Chen. So thanks, everybody. Thank you for being here. We will adjourn the meeting, and thanks very much to our speakers. And
thanks,
Steve
everyone.
reminds me, we'll have YouTube on the video will be on YouTube eventually in some edited form.
Yep.
Thanks, Patty.
Thank you.